Good evening, everyone. Today we are in the week 5 problem solving session for the NPTEL course, NMR Spectroscopy for Structural Biology. So I hope all of you have gone through the week 5 lectures. So in the last week, we were looking into different aspects of solvent suppression methods. And then we had also talked about chemical exchange rates. How can we determine the exchange that is happening between chemical exchange happening between the domains of a protein. So, for example, if a domain is of a protein is moving from a coiled state to an uncoiled state, or from a helix to a coil, how such experiments are performed. Then we had also seen selective population inversion experiments. So, in this session, we will talk about 2D NMR experiment. So, we will revise the concepts that were discussed in 2D NMR experiments, starting from what is a 2D NMR experiment, and then the 2D NMR experiments like COSY and TOCSY experiments. So we will wait for two or two more minutes and then start today's session. So we will quickly go through the concepts of 2D COSY experiments that are related to two-spin system, three-spin system. So AMX spin system, if the AMX linear spin system and AMX triangular spin system. And we will also revise the concepts that were discussed in TOCSY experiments as well. And also the magnetization transfer experiments, like the different spin systems that arise because of the magnetization transfer, uh, starting from single quantum coherence, double quantum coherence, zero quantum coherence. So let's wait for another 1 minute and then we can start the class at 7:05. Okay, so I'll start presenting the slides for today's session. So I hope the slides are visible. Okay, so yeah, good evening everyone. So I'm Akash Jyoti Pathak. I am a PMRF TA for this NPTEL course, NMR Spectroscopy for Structural Biology, and today we are in the week 5 problem solving session for this course. So the course is a rerun course, and it has been taken by Professor Ashutosh Kumar and Professor Ramakrishna Hosur sir. So let's quickly revise the concepts that were discussed related to the 2D NMR experiment. So the limitations of what are the limitations? So why is 2D NMR experiments required? So it is because of the limitations that is there in case of 1D NMR experiment. So what is the limitation in case of 1D NMR experiment? If we see that in the conventional FT Fourier transform NMR experiment, what are we doing is we are collecting the FID, right? So the FID that is collected, that is in the time domain. And then we do a Fourier transform so that this time domain can be converted into a frequency domain. Because when we look at the spectra, NMR spectra, we see that the frequency of the chemical shift is present on the x-axis and the intensities are present on the y-axis. So after a Fourier transformation, we can convert this time domain signal into a frequency domain spectrum. So in case of 1D NMR, what we are seeing is that we are only having one frequency axis, right? So this is taken from the lecture slide. So as you can see here, that this is the 1D proton NMR spectra of a protein called lysozyme. So here you can see that there is only one axis, one frequency axis. Okay. And because of this, what is happening? So if we consider the protein lysozyme, so it is a 15 kilodalton protein. So it has around 130 amino acid residues. So and 130 amino acid residues will give rise to around 400 protons. So because of such large number of protons, what will happen is that everything will appear overlapped, right? So you will not be able to deconvolute that which peak is arising from which proton in the protein lysozyme, right? So because in the proton you can as we had seen in the previous lectures as well, the proton spectra spans from 0 ppm to 11 ppm, right? So everything all those 400 protons will appear overlapped in this 1D NMR spectra. So the limitation is that for small molecules, proton NMR or 1D NMR experiments are manageable, in the sense like we had seen how we can identify the protons which is present in vinyl chloride or in ethanol, or small molecules like which are having only 2 or 3 carbons and having like 7 or 8 protons. So for such type of small molecules, proton NMR can give us can uh, can provide information, or we can deconvolute the peaks and understand that which peaks are arising from which protons. So for small molecules, it is manageable. But for large molecules like proteins or nucleic acids, which are having hundreds of protons, the resonances will overlap. And so, yeah, so there will be many resonances, and these resonances will overlap with each other. And as a result, we cannot identify or we cannot assign the individual peaks separately. Okay, and also determining the coupling constants become extremely difficult. So the overall message from this slide is that the 1D NMR experiment becomes difficult for large molecules because many resonances occupy the same chemical shift dimension. So that is why the NMR 2D NMR experiments were developed. So we can see the difference, or what was employed in 2D NMR experiment is that now instead of having instead of having a single frequency dimension, as was the case in 1D NMR, we are now having two frequency dimension, that is F1 and F2, or T1 and T2. So So yeah, so instead of spreading the resonances over one frequency dimension in 2D NMR, what is done is the resonances is spread over two dimensions, F1 and F2. So this second dimension allows us to establish the relationship or correlations between different resonances. So you can see how. So here you can see that how the two in a 2D NMR there are four periods actually. So first is the preparation period, followed by an evolution period, then a mixing time, and followed by the detection. Okay, so there are basically four periods. So the preparation period is the period where we can the spin systems are prepared. So yeah, either the spin systems can be in a steady state, or they can be they can they can be in a different equilibrium state. Then in the evolution time, what will happen after the RF pulse will be provided, and then the spins will start precessing according to their own frequency. So the spins will evolve according to their frequency. Then in the mixing time, what will happen? The magnetization transfer between the spin systems will take place. So the mixing time allows magnetization transfer or correlation. And during the T2 period, what will happen? The FID is collected. Okay. Okay, the FID is collected during the T2 time period. So what is done is that in a 2D NMR, there is a small increments in T1 is made. And at each increment of T1, FID is collected with each increment of T1. Okay, so basically what is happening in 2D NMR is an additional time period. T1 is introduced. And this T1 is made in. So if you see this figure, this is from the lecture slide. So there is an increment. So at each cycle, there is an increment in the T1 period, and with each increment in T1 period, the FID is collected with each increment in T1 period. Okay, so what will happen is, so yeah, T1 is called the evolution period, and T1 it is also called as the indirect. It is also called as the indirect detection dimension. Why is it called the indirect detection dimension? Is because the FID is not collected during the T1 period. The FID will be collected in the T2 time period, but any information that is present in the T1 will be when we collect the FID, that information will be present in the T2 dimension. So a series of experiments are performed with increments of T1, and an FID is collected for every value of T1. So as a result, what we will have is we will have multiple FIDs with each being a function of T2. But the amplitude or shape of each FID will depend on T1 because the spins evolve during T1. Therefore, the signal now becomes, so the signal will depend on both T1 and T2, right? So this is a T2, a 2D time domain signal. So what is happening in the T1 period is that during the T1 period, the spins will evolve or precess according to their own frequency, and this evolution will encode the frequency information. So the subsequent FID therefore carries information about what happened during the T1 period. Okay. So yeah, so this is the important point, that the amplitude or shape of each FID will depend on T1, and so when we will perform a series of experiment with the increment of T1, we will have multiple. So at every T1 value, we will have multiple. So we will have an FID, a corresponding T2 value. So we will get multiple FIDs, and then we can deconvolute it, and we can get the information, which is so we will have two axes, the F1 axis and the F2 axis, and we can have the information like what is the info for a particular spin, what is the information in the F1 dimension, and what is the information in the F2 dimension. So what is happening in the mixing time? So in the mixing time, what happens is that the magnetization transfer takes place from one spin to another spin. So mixing connects the information encoded during T1 with what is observed during T2. Okay, so it is connecting the T1 with the T2. So the magnetization can be transferred from one spin to another spin. So during that mixing time period tau m, what will happen? The magnetization transfer can either be complete or it can be partial. So we had seen some of this concept in the NOE that we were discussing in the last week. So polarization transfer, for example, when in we had seen that in case of NOE, partial transfer is allowed. The tau time period, the mixing time period is selected in such a way that only a partial magnetization will be transferred from one spin to another spin. So the mixing establishes correlations between frequencies associated with the T1 and T2 time period. So we have now we have information in both T1 and T2, so we need to do a 2D FT Fourier transform. So when we do a two-dimensional Fourier transform with this time domain, this T1 domain will get converted into F1 and T2 will get converted into the F2 frequency domain. So F1 will be the frequency information which is present during T1 and F2 is the frequency information which is detected during T2. So T2 is called the direct detection. Why is T2 called the direct detection? Because the actual FID is directly recorded by the spectrometer during the T2 period. So actual FID is directly recorded by the spectrometer. That is why T2 is called the direct dimension, or F2 is called the direct dimension or direct detection dimension, because it is during this period the FID is collected, and F1 is called the indirect dimension or indirect. So during T1, no FID is directly collected. That is why T1 is called the indirect detection dimension. So instead, the effect of evolution during T1 is encoded into subsequently acquired FIDs. So your T1 is called the indirect detection and T2 is called the direct detection. Okay. So this figure was there in the lecture. So what is 2D NMR actually doing is that 2D NMR introduces an additional evolution period T1, and then it systematically increments this T1 period and records multiple FID. And then these multiple FIDs are then Fourier transformed into both T1 and T2 dimension to generate two frequency dimensions. Okay, so here you can see that this is your T1 dimension and this is your T2 dimension. So in the T2 dimension, you are collecting the FID, right? So with each increment of T1, you are collecting FIDs. So with each T1 increment, you are collecting one FID. So suppose you collected, this is your first FID. So when you Fourier transform this first FID, you get this type of signal. Then this is your second FID. Again, you Fourier transform it. You see the signal is reduced in case of the Fourier transform of the second FID. Then this is your third FID. Here the signal has become zero, right? So when you Fourier transform it, you get zero signal. Then this is the fourth FID. When you Fourier transform it, your signal decreases. It becomes negative. So now with all the combining all these information, the final T2 or the free F2 in the F2 dimension, when you Fourier transform it, you get this type of signal. Okay, so now if you like convert this F2 frequency information into F1 frequency information. So at each point, like at A point, so if you see that at this point, A point, you can see that at the A point, this is zero. This in this FID also it is zero. The frequency is zero. Here also it is zero. Here also it is zero. So that is why. If you do a Fourier transform of this, you will get the zero signal. Then coming to the B point. So at the B point, at each of the FIDs, if you look here, B point, you can see that there is a slight decrease in the FT in the there is a slight dip in the signal of the Fourier transform signal. So when you again deconvolute it, you will get a Fourier transform of this signal will give you a small positive peak. Then at point D, so at point D, you can see that there is a, if you combine all these frequencies at point D, you get a very sharp dip in the signal in the FID. So when you Fourier transform it, this will give you a this type of signal. So at each point, if you like at C point, at D point, at E point, F point, G point, if you combine all these frequencies at each of these points, then you can actually get the information that was present in the T1 dimension, right? So so now you are having two types of information here. You are getting information which is present in the, so your your Fourier transformation is giving you information in the F1 dimension and in the F2 dimension. So you can plot this and you can have both information in the F1 and F2 dimensions. So now that was the basic principle of 2D NMR. So now another concept that was discussed was how this 2D NMR is now applied to J-resolved spectroscopy. So in 2D J-resolved spectroscopy, what you are trying to do is you are trying to separate the J coupling information with the so in 2D J-resolved spectroscopy, the basic idea is that you are separating the J coupling information from the chemical shift information. Okay. So for a particular spin system, you are decoupling or you are separating the J coupling information that is present in that particular proton or particular spin system from the chemical shift. So in very simple terms, what it means that your F1 dimension will contain the J coupling information, and your F2 dimension will contain the chemical shift information. Okay, so in a 1D spectrum, what is happening that each resonance can be split by J coupling, right? We had seen this that in the n+1 rule, we had seen that if the proton is nearby to the neighboring the environment, what is the chemical environment that is present of a particular proton, that the neighboring proton can split that particular proton into doublets or triplets or doublet of a doublet. So each resonance can be split by J coupling into doublet, triplet, doublet of doublets, and this can make the spectra complicated, particularly for larger molecules. So the goal of J-resolved spectroscopy is basically to separate the chemical shift from J coupling onto different dimensions. So basically, this is how the pulse sequence looks for a 2D J-resolved spectroscopy. And if you look at this pulse sequence carefully, then you will find that it is having the same sequence as that of a spin echo pulse sequence. So it is having the same 90 tau 180 90 tau 180 tau pulse sequence. So what is happening is that, so this is essentially a spin echo during the evolution period. Evolution period is your T1 time period, is called the evolution period. So your evolution period is having the spin echo sequence. So in spin echo, as we had learned in the last lecture, that in the last session also we had revised the concept of spin echo, that in the spin echo the chemical shift evolution is refocused, but the J coupling evolution is not refocused. Therefore, during T1 only J information will evolve. Okay, so because this is a spin echo sequence, and the basic idea of a spin echo sequence is that the chemical shift evolution will be refocused, but the J coupling evolution is not J coupling information is not refocused, and it will evolve. So your basically your F1 dimension will have the J coupling information. So during T1, what is happening? Your chemical shifts are refocused, and therefore your F1 dimension will provide you information about the J coupling. And during T2, normal acquisition occurs. So your T2 during your, yeah, so that is what has been shown in this figure as well. So your spin echo, your chemical shifts are refocused, while in this T2 dimension you are collecting the T2 dimension, you are having the chemical shift information, and you're in the T1 dimension you are having the J coupled information. Okay. So this is how a J resolved 2D J resolved spec J resolved spectroscopy looks like. So your F1 dimension will have the J coupling information. Okay. And your F2 dimension will have the chemical shift information. So you can see here in this figure that in your F1, your F1 dimension, there are multiple multiplets. The multiplets can be observed as characteristic patterns in the 2D spectrum. Now, what happens is that initially the spectrum is oriented at 45 degree. Okay, and then during processing, what has to be done is that these peaks that are arising, they have to be shifted or they have to be moved so that the multiplets becomes aligned. Okay, so this is so now when you take a contour map, or when you take a convert this into your 1D NMR will look like this signal. So here you are having two peaks. So this is giving you this type of signal, and here you are having three peaks. So this is giving you a sharper peak. So this is called as broadband decoupling. So this effectively separates the two types of information. So basically, what you have to remember here is that your F2 dimension is providing you the chemical shift information, and your F1 dimension is providing the J coupling information. So your 2D J coupled spectroscopy is allowing you to effectively separate the two types of information. Okay. One type of question related to. So let's try to solve this question, which is related to 2D NMR. So in a 2D NMR experiment, which of the following statement is correct? So first option is data is explicitly collected during two independent time variables T1 and T2. Data is explicitly collected only during T2. Data is explicitly collected only during T1. The spectrum is generated by frequency selective excitations along the two frequency axes F1 and F2. So just now we saw that your 2D NMR experiment looks somewhat like this. It has a preparation time period, right? Your preparation time period is followed by an evolution time period T1. Then there is a mixing time, which is called tau m, right? And following this mixing time, there is the T2 time period where the actual FID is collected. So your T2 is called the direct detection. It's called the direct detection period, and this is your indirect detection period. So we saw this concept through that figure as well. So you collect multiple FIDs, and how when you deconvolute each of the frequency which is present in this direct detection time period, you can get information of the frequency information that was present in the T1 time period. Okay, so what will be the answer for this question? So the data is explicitly collected during T2, right? That is why it is called the direct detection period. So answer is option B, right? So in a typical 2D NMR experiment, there are two time variables involved. Just now we saw that is T1 and T2. T1 and T2. So the evolution of the spin system during T1 leads to encoding of frequency information along the indirect dimension. But data is not explicitly corrected during the T1 period. right? Instead, T1 is incremented systematically, and the experiment is repeated for each increment, ultimately encoding this time dependence in the recorded data. So the actual data collection takes place during which period? T2. That is why it is known as the direct detection period, and this corresponds to the F2 frequency axis. So your data is explicitly collected only during T2, and the T1 evolution information is encoded into the signal by varying T1 between acquisition. Okay. So answer this second question. Fourier transform of a complex NMR signal ST1, T2 leads to. Okay, so let's discuss the concept of magnetization transfer first. Okay, because this question is related to that concept. Then we can discuss this question. Okay, so after this lecture, we the concept that was discussed was the magnetization transfer or magnetization flow. That is what will happen during the evolution, during the. So your magnetization transfer or magnetization flow will take place during which period in the 2D NMR experiment? So it will take place during the mixing time, tau m time period, right? So during a multidimensional NMR experiment, magnetization can move from one state to another during different periods of the pulse sequence. So during different periods of the pulse sequence, there can be transfer of the magnetization, and the different evolution and mixing periods will determine how magnetization is transferred. So what is your Tm and what is your tau m values, that will determine how the magnetization is transferred. So for a spin K, there can be three basic magnetization terms, right? So a spin K can have three basic magnetization terms. We had this was discussed in the lecture. One is I. It can be IKX, IKY, and IKZ. Right. So there can be three basic magnetization terms. So your IKX and KY, these are your what? These are your transverse magnetization components, right? Because XY axis, because they're present in the XY axis. So the magnetization which is present in the XY axis, they will be called as the transverse magnetization components, and your KZ is what? It is the longitudinal magnetization. Longitudinal magnetization component. Okay. So a spin K can have three magnetization components: IKX, IKY, and IKZ. Your IKX, IKY are your transverse magnetization component, and IKZ is the longitudinal magnetization component. So anything that is present, any magnetization, if it is present in the transverse axis, that can be detected because your detector is present in the XY plane. So depending whether it is present on the X axis, we have seen that these concepts in the quadrature detection lecture when we were discussing lecture 2 or lecture 3. So your transverse magnetization are the only observable. So NMR can only detect the transverse magnetization. Therefore, only IKX and IKY are observable, but IKZ is not observable. So any component which is present in the Z axis or in the longitudinal axis, that cannot be detected. That is not observable. So the detector is sensitive to magnetization in the transverse plane. Therefore, Z magnetization must first be converted into. So if you want to detect the information which is present in the longitudinal axis, that has to be converted into the transverse magnetization before detection. Okay. So these are the three basic magnetization terms that is possible. Okay. So this was explained in the lecture as well. So you can see that when you do when you are collecting an FID in the x-axis and you do a Fourier transform, then what is present? You get a doublet, or you get a doublet of K, which is also it is an in-phase doublet. So suppose spin K is coupled to another spin L. Okay, so your spin K is coupled to another spin L. Then what will happen? So you have two spin systems, KL, and if K is coupled to L, then what will have? There will be peak splitting. Or L will result in the splitting of the peak of the K spin system. So you will get a doublet. You will get a doublet. Okay. So when you do a Fourier transform, you will get the in phase doublet signal. Now, depending on where your detector is present, so if your detector is present in the x-axis, you will get in phase or absorptive signal, right? In phase doublet signal, and the y-axis will give you dispersive signal. While if your detector is present in the y-axis, then when you do a Fourier transform, you will get you will get vice versa signal. You will get in phase doublet signal of the in the y-axis, and in phase dispersive signal in the x-axis. Okay, so this is interchangeable depending on where your detector is present. So suppose spin k is coupled to another spin I, the kx magnetization will produce a multiplet. Okay, so if we consider a very simple spin system like KL, then you will have a doublet for K and you will observe a doublet for L. So your K spin will result in peak splitting of the L spin, and your L spin will result in peak splitting of the K spin. So you will get two doublets in your spectra. Okay, so now depending on when you do a Fourier transform and where your detector is present, either you will get a in phase absorptive signal, or you will get a in phase dispersive signal. So this is how your IKX and IKY signal looks like. So when you do a Fourier transform, but other than this IKX and IKY signal, you can have this type of magnetization terms as well, like 2 IKX ILZ. So what does this mean is that this is X magnetization of K. So if you have anything like this, something, if you get these terms, so when you actually this is a course for structural biology, right? So very detailed calculation as to how we will get these type of terms are not there in this course. So quantum mechanical calculations, when you do those Fourier transform analysis and everything, how we get this type of term, that is not in the preview of this course, but we can get some terms like this during the magnetization transfer. So what does this mean is that it will mean it is xkx, right? So it means x magnetization of k, x magnetization of k, which is antiphase, because you have lz, right? Lz, that is why it is, which is antiphase with respect to l, antiphase with respect to l, with respect to L, with respect to spin L. X magnetization of spin K, which is antiphase with respect to spin L. So you can have several antiphase magnetization terms. So what are these antiphase magnetization terms? So we looked at terms like 2IKX, I, L, Z. So this is X magnetization of K, antiphase with respect to spin L. You can have terms like 2I, KY, I, L, Z. So this would mean Y magnetization of spin K, antiphase with respect to spin L. Or you can have terms like this also, 2I. So this was your X magnetization of K and Y magnetization of K. Similarly, you can have X magnetization of L, which is antiphase with respect to K. So this will be KZ. Or you can have terms like I, K, Y, ILY, ILY, IKZ. Okay, ILY, IKZ. So this will be Y magnetization of L, antiphase with respect to spin K. So these are the antiphase magnetization terms. So your antiphase signals will look like what? It will have a positive component and a negative component. So these are your, when you do a Fourier transform, you will get a positive component and a negative component. So these are called antiphase signals. Okay. So for this, like for the K spin, you can have X magnetization, which is antiphase with respect to L. You can have Y magnetization of K, antiphase with respect to L. You can have X magnetization of L, antiphase with respect to Z, or you can have Y magnetization of L, antiphase with respect to spin K. Okay. So now these antiphase and in-phase terms, they are actually arising because of this these, spin, systems we have, right? So in this two spin system, we have four states, right? For a two spin system, if you see, we have four spin states: alpha alpha, this is beta beta, this is alpha beta, and this is beta alpha. Okay, so if there is a transition from alpha alpha to alpha beta, or from alpha alpha to beta alpha.
Sir, can you go to the back page?
Yeah. Yeah.
1 minute, sir. Okay, sir. Okay, continue.
Okay, so if the spin systems are there is a transfer from the alpha alpha spin state to the alpha beta spin state, or there is a transfer from alpha alpha to the beta alpha, or beta alpha to beta beta, or alpha beta to beta. So these are all what? So this is actually m equal to -1. This is m equal to 0, and this is m equal to +1. Okay, so these transitions, alpha alpha to beta alpha, or this single state transfer, is called as the single quantum coherences. So single quantum coherences are those that would result in a delta m of 1, right? So either it can be +1 or it can be -1. So either this alpha alpha. So if we consider two spin systems, right, L and K. So if your L is in the alpha state and K is also in the alpha state, so these things we had seen already, right? So if anything in any transition is resulting in a delta M of ±1, then that is called as single quantum coherence. Okay. So single quantum coherences would mean transitions from alpha alpha to alpha beta, or alpha alpha to beta alpha, right? Alpha alpha to alpha alpha to beta alpha, or there can be alpha beta to beta beta, or there can be beta alpha to beta beta. So these are called the single quantum coherences. Similarly, there can be transitions that result in double quantum coherences. So double quantum coherences are these ones, alpha alpha to beta beta. So because here the delta m would correspond to what? It is like -m minus of 1. So it is like 2. So the delta m is ±2, either alpha alpha to beta beta or beta beta to alpha alpha. So these transitions will result in double quantum coherences. So transitions with delta m equal to ±2 are called double quantum coherences, and transitions with delta m equal to 0. So if there is a transfer which is taking place between this alpha beta and beta alpha, right? Alpha beta and beta alpha, then these are called the zero quantum coherences, because here the magnetization is 0, m is 0. So that is why these are called the zero quantum coherences. So one thing that you have to understand, so these are these concepts come from the quantum mechanical calculation, but for this course, what is required to be understood is that there can be three types of transitions, or there can be three types of coherences, or three types of coherences can be obtained. One is the zero quantum coherence, which is because of delta m equal to 0. There can be single quantum coherences, which is because of delta m equal to ±1, or there can be double quantum coherences because of delta m ±2. So the important point over here is that the single quantum coherences are observable. So these are observable, while these zero quantum coherences and double quantum coherences, these are not observable. These are not directly observable. Not observable. Not directly observable. So if you want the information which is present in this zero quantum coherence and double quantum coherences, you have to convert it into single quantum coherences, and then the signal can be, or then the information can be decoded. Okay. So now what happens is this DQC and ZQC, double quantum coherences and zero quantum coherences, have terms which are combination of this single quantum coherences term. Okay. So when you are having terms like 2 IKX ILY, 2 IKY ILX, so these indicate mixture of zero quantum coherences and double quantum coherences. Okay, so here you can see in this figure, this is from the lecture slide. So just look at this figure. So you are having is alpha alpha, it is m equal to 1, beta beta is m equal to -1. So these are your single quantum coherences. Alpha beta to beta beta, alpha alpha to beta alpha, these are single quantum coherences. Alpha beta to beta alpha is your zero quantum coherence, and alpha alpha to beta beta, these are your double quantum coherences. So these terms that you are getting, so basically this ikx, iky, ikz, these are your single quantum coherence. These magnetization terms that you get, these are because of single quantum coherences. While if you have terms like 2ikx, ilz, right? So this was X magnetization of spin K, antiphase with respect to spin L. So these are mixture of zero quantum coherences and double quantum coherences. And when you are having combinations of these terms like 2ikx ILY plus 2iky ILX, so combination of these terms can give you pure double quantum coherence systems or pure zero quantum coherences. So if you get an expression like 2ikx ILY plus 2iky ILX, this will give you pure double quantum coherence. And if you have 2ikx ILY minus 2iky ILX, this will give you pure zero quantum coherences. So this was what was explained. So if you have terms like ikx ILY, iky ILX, or ikx ILX, okay. So these terms, so these are IKY, ILY. These are indicating combinations of double quantum coherences and zero quantum coherences. Okay, one thing that I mentioned, I, I, I I made a mistake while saying one thing. So this 2 IKX, ILZ, these are also your single quantum coherence system. Okay, IKY, ILZ, or the terms that we discussed in 2 slides before. ILX, IKZ. So these are your single quantum coherences. But if you have terms like this, your when your spin L is in Y magnetization, or your spin is in X magnetization. So your ILX, ILY, ILX. So these terms, these are indicating double quantum combinations of double. These are having mixtures of both double quantum in coherences and zero quantum coherences. Okay. So other than this zero quantum coherence, double quantum coherence, so these are these are your together they are called as the multiple quantum coherences. So single quantum coherence, double quantum coherence, zero quantum coherence, and triple quantum coherence, they are collectively called as the multiple quantum coherences. However, the detector, so what you have to understand is that the detector can observe only single quantum coherences. So therefore, during an experiment, what has to be done? So during an experiment, these states can occur. This single quantum coherence, DQ, double quantum coherence, or zero quantum coherences. These states can occur as part of the magnetization transfer pathway. So during the magnetization, so this is how it was explained in the lecture. So this magnetization, once it is applied, it can have in the T1 period, it can have at a particular time, it can have SQC. Then there can be DQC, ZQC, but ultimately the magnetization has to be converted into single quantum. Ultimately, for the detection, all these signals has to be converted into single quantum coherences to be for the detector system. It has ultimately all these signals has to be converted into single quantum coherences. Okay, so the important point is that all these multiple coherences term like zero quantum coherence, DQC, triple quantum coherences, these are not directly measurable in the detector system, but they can appear in the magnetization flow. So during the flow, magnetization transfer or magnetization flow, these terms can appear, and they have to be converted into single quantum coherences for the signal to be detected. Okay. So there can be a 3-spin system like AMX spin system. So for a now 3-spin systems can have there. So there can be 3 spins like L, K, KLM, KLM. So earlier we were considering 2-spin system. So we were having 2 spin. One was L spin and another was L spin. So there can be 3-spin system like K spin, L spin, and N spin. So So that was so this is what explained in that. So if you are having terms like 2IKZ, ILZ, so these are two spin ZZ ordered. So they have to be converted into SQC or DQC or ZQC. Okay. So suppose we are having now three spins like K, L, and M. So these individual, so now these three spins can have individual spin magnetization IKX, ILX, or IMX. So we can have two spin terms like 2IKX, ILZ, or 2IKX, IMZ, or we can have three spin terms. So these are your three spin terms for IKX, ILY, IMZ. These are your three spin terms. Okay. So if you are having terms like this IKX, ILX, IMX, these are X magnetization. What does this indicate? It indicates X magnetization of spin K, X magnetization of spin L, and X magnetization of spin M. Now, if you are having terms like 4 IKX, ILY, IMZ, then this would mean X and Y magnetization of spin K and spin L antiphase with respect to spin M, right? Or we can say that DQ and ZQ, double quantum and single quantum of K and L spin antiphase with respect to spin N M. But if you are having terms like IKX, ILZ, IMZ, then this would mean single quantum of spin K. Okay, single quantum of, so you are having only the K spin which is in the X magnetization. So that is why it is single quantum of K antiphase with respect to both L and M spin. Okay. So now if your spin systems are coupled to each other, okay, your K spin, L spin, and M spin, they are coupled to each other. And if you do a Fourier transform, then what will you get? You will get a doublet of a doublet, right? Because your L spin will cause splitting of the spin K, and your M spin will also result in splitting of spin K. So you will your L spin will split your K spin into doublet, and this doublet will again be split by M spin into another doublet. So that is how you will get doublet of a doublet. So this is how your doublet of a doublet spectra looks like. Then so this is how your IKX, if you do a Fourier transform of this IKX, you will get this type of signal frequency information. Then if you if you have this term 2IKX, ILZ, and if you collect the FID and do a Fourier transform, what you will get? You will get, so this is X magnetization of spin K antiphase with respect to spin L. So you will get antiphase terms plus minus, right? And then if you have these terms like 4IKX, ILZ, IMZ. Okay. So here what will happen? Here, if you do a Fourier transform, you will get these signs will be reversed now in this case. So this is doubly, so this is actually case of a doubly antiphase magnetization. Okay, because this K spin is antiphase with respect to both spin L and spin M. So this is doubly antiphase magnetization. Then you can have terms like IKX, ILY, IMX. So this is a case of triple quantum coherence. KX, your K spin is X magnetization of spin K, Y magnetization of spin L, and X magnetization of spin N, M. So this is triple quantum coherences, these terms. Okay. So this question we discussed. So answer this question. Fourier transform of a complex NMR signal ST1 Q2 leads to absorptive line shapes along both frequency axis, dispersive line shape along both frequency axis, absorptive line shape along F1 and dispersive line shape along F2, or there can be mixed line shape along both frequency axis. What will be the answer? So basically here you can get all different types of information. Like we will see in case of 2D COSY experiments also, that you can have both absorptive line shape and your dispersive line. You can get mixed line shape along both frequency axes. So in 2D NMR, after performing Fourier transformations along both the time variables T1 and T2, the resulting spectrum can have a mixed phase. So this means that it can have both absorptive and dispersive components. and they can be present along both. They can these absorptive and dispersive line shapes can be present in both F1 and in F2 as well. Okay, so the combination of these real and imaginary components corresponding to absorptive, real means absorptive and imaginary means dispersive. Right, we have seen that cos, if there are cos terms, cosine terms, it gives us absorptive signals. If there are sine terms, we get dispersive signal. So this leads to mixed line shapes which can appear in both dimensions of the spectrum. So we will get mixed line shape along both frequency axes. So the resulting peaks might show different phases depending on the method of detection and the processing. So whether, as we were discussing, whether your detector is placed in the x-axis and your FID is in the x-axis, then you will get absorptive doublet signals, and in the f-axis you will get dispersive signals. So answer is option D. So in a homonuclear 2D J-resolve spectrum, so answer this question we discussed just now. So homonuclear 2D J-resolve spectrum will have what? F1 axis has chemical shift, F2 axis has coupling constant. F1 axis has coupling constant, F2 axis has chemical shifts and coupling constant. F1 axis has coupling constant and F2 axis has chemical shifts. And option D is F1 axis has chemical shift and coupling constants. What will be the answer? So answer is option C. We just now saw in a 2D J-resolve spectrum, the pulse sequence is designed in a way that your F1 frequency dimension will give you the information about the J coupling, and your F2 dimension gives information about the chemical shift. So in a 2D J-resolve spectrum, the NMR spectrum, the F1 dimension displays coupling constant, while the F2 dimension displays chemical shift. This experiment effectively separates the coupling information and the chemical shift information into two different dimensions, allowing for easy interpretation of the complex spectrum. Okay, so this point is clear, right? Few questions related to this. There were a few questions related to the concepts that we discussed just now. Let me add those questions quickly. Just give me a moment. So I've added the question. So there were a couple of questions asked in the assignment. So so this was one of the questions in the multiples 2D experiment. Which of the terms are observable? So we just now discussed that the single quantum coherences are observable, right? And if you have spin systems like IKX, IKY, IKZ, these are single quantum coherence systems. So answer is this IKX. Which of the terms are observable will be IKX, option A, right? Then yeah, then there was another question like 2 IKX ILY represents. So if you are having terms like 2 IKX ILY, or you are having terms like 2 IKY ILX, or there are like XY terms too. So these terms will basically, what do they represent? They represent mixture of double and zero quantum coherences, and combinations of these terms can give you pure double. So if it is 2 IKX ILY plus 2 IKY ILX, it will give you what? It will give you pure double quantum in coherence terms, and if there is a minus between these two terms, it will give you pure zero quantum coherence terms. So answer is option D for this question. So 2 IKX IL1 will represent mixture of double and zero quantum coherence. So in the multipulse experiment, the selection of pathway should end in. So we just now learned this concept, right? So multipulse experiment. So whether you are so during the magnetization flow or magnetization term transfer, you can have ZQC, DQC. These different terms, terms can arise, but ultimately during detection, for detection, all these signals must be converted into single quantum coherence term for the detector to detect the signal. Okay, so it should end in a single quantum coherence. Okay, so we had seen in this slide as well. Okay, so ultimately the magnetization must be converted into the single quantum coherence for detection. Okay, so these points are clear. Now we will quickly discuss the 2D COSY experiment. So what is a 2D correlation spectroscopy experiment? So this was discussed in quite a great amount of detail. So what happens in a two-dimensional correlation spectroscopy experiment is basically in such type of NMR experiments we get two types of peaks. One is the diagonal peak. So you can see here in this figure as well. So these are called your diagonal peaks. These are your diagonal peaks, and these are your cross peaks. These are your cross peaks. Okay, so we get two types of peaks in case of a COSY experiment. One is the diagonal peaks and another is the cross peaks. So basically, your in each dimension, in your F1 dimension and F2 dimension, so your F1 dimension has 1D spectrum, and your F2 dimension will also have this 1D spectrum. Okay, so so you are trying to find the correlation. So basically, if your COSY experiment will basically give you information about the J coupling between two spins. So the diagonal peaks, when will we get the diagonal peaks? So the diagonal peaks will occur when the frequency is same along both the dimensions. Okay, so that is why A is correlated. So your diagonal peak is, if you see, you will get that your A is correlated to A and your B is correlated to B. So you are getting a diagonal peak. Um, so each spin is correlated to itself, but the cross peaks will occur off the diagonal. So away from the diagonal, the peaks that you obtain is called the cross peaks. So the cross peaks are information carriers in the 2D correlation spectroscopy. Okay, so if you get a cross peak, in a COSY experiment, then it would suggest that your A spin is coupled to the B spin. Okay, that is why you are getting a cross peak. But if your A spin is not coupled to the B spin, then you will not observe this cross peak. So basically, your cross peaks are the information carriers, and your diagonal peaks are they are just that each spin is correlated to itself. So that does not provide us any essential information. Okay, so in a 2D homonuclear COSY experiment, we get diagonal peaks and cross peaks. So the diagonal peaks are found along the diagonal of the 2D spectrum. So they arise when there is no transfer of magnetization. So if there is no transfer of magnetization, if there is no transfer, that means that the two spin systems are not coupled to each other. So there would be no transfer of magnetization. They represent autocorrelation. That is, each spin is correlated with itself. So the diagonal peaks are related to the chemical shift of the nuclei and do not involve any magnetization transfer mechanisms like dipolar or J coupling interactions. So J coupling interactions like are the magnetization which gets transferred through bonds, and dipolar interactions are the magnetization transfers which takes place through space, as we had seen in case of NOE. NOE, that the magnetization transfer was taking place through space, and J coupling is through bonds like 3-bond coupling, 2-bond coupling. So diagonal peaks are essentially the 1D NMR spectra. Really. It has there is no other information, but the cross peaks in a COSY spectrum will arise due to transfer of magnetization between two different nuclei through scalar coupling, also known as J coupling. So in 2D, in a COSY experiment, the cross peaks will arise mainly because of J coupling. Okay. If they are coupled to each other, then there will be magnetization transfer, and then only we will see the cross peaks arising. So this J coupling interaction mediates the transfer of magnetization between coupled spin, creating off diagonals or cross peaks in the spectrum. Okay. So basically, why what extra information are we getting in case of a 2D NMR experiment or 2D COSY experiment is that in 1D NMR spectrum, 1D spectrum, we are only getting the information about the chemical shift of a particular spin, right? So it may tell us that there is a signal at this chemical shift, but in a complex protein spectrum, we would want to determine which spin is coupled to which particular spin. So whether these protons that are present, they belong to the same amino acid or they are part of different amino acid, which resonances are connected. So all these informations are not provided by a 1D NMR experiment. So a 2D correlation experiment will provide information on these by producing cross peaks. So a COSY is a 2D NMR experiment which is used to identify J-coupled spin. So this is important because COSY will provide information about what is the J coupling between the spins. So here also what is done here, first of all, we give a 90-degree pulse, and this is called the preparation period that we were understanding. So this is the preparation period. Then there will be a T1 evolution period, right? So T1 evolution period, what will happen? The spin will precess according to its own frequency, and then a second pulse can be provided. Okay, and then after, so this is called the mixing time, and then there will be detection to T2 detection. So you can see here, it was explained in the lecture. So you are having a spin K spin, and this K spin is oriented in the Z direction. So initially it was IKZ. Now we have provided this 90-degree pulse, and now your K spin has oriented to the Y axis. So now you are having the magnetization in the Y axis. So that is why it has been labeled as IKY. And now after the mixing time, a part of this magnetization, a part of the K magnetization can be transferred to the L magnetization, to the L spin. So a part of the K magnetization can get transferred to the L spin. So now in the T2, we will get both K and L magnetization. Okay. So in case of, if we look how a COSY spectra will look for a particular amino acid. So that this was explained with the example of an amino acid valine. So if we look at the structure of the amino acid valine, then you can see that its R group, the side chain has this CH3 C. So this alpha carbon is bonded to a beta carbon, and then this beta carbon is again bonded to two gamma methyl groups, right? CH3, CH3. So when you do a when you take a 2D COSY spectra of this amino acid, of this valine amino acid, what you will get? So here you will get a, so you are having how many protons are there? So you are having this NH proton, amide proton. Then you are having this alpha carbon proton, right? Alpha carbon proton. Then you are having this beta carbon proton that has been labeled as H beta. And then you are having two gamma protons, right? CH3 and this CH3. So these are two gamma carbon protons. So you can see your 1D, your diagonal peak is your diagonal peaks are basically your 1D NMR spectra. Basically, it is the 1D NMR spectra. So you will get four different proton peaks. One for the amide proton, one for the alpha proton, one for the beta proton, and two for the gamma protons. Okay. Now your correlation, your in your COSY, what is in a very simple term, what it is trying to say is whether this amide proton is coupled to this alpha carbon proton. So if it is coupled to alpha carbon proton, then you will have a cross peak arising here. So this is a cross peak arising over here, and the same will be obtained here as well. Okay. It means that this amide proton is coupled to your alpha carbon proton. Similarly, if your alpha carbon proton is coupled to your beta carbon proton, then again you will have a cross peak over here. Okay, cross peak over here. And the same will be present on other side of the diagonal as well. Now, if your beta carbon is coupled to your gamma carbon, you will get a cross peak over here. So here you see there you are getting two gamma proton because your beta proton is coupled to your gamma proton. You are getting two cross peaks that is present over here and that is present over here. So basically, when you perform these type of experiments and combination of this COSY, NOSY, TOCSY, all these different experiments, once you have performed, you can assign the peaks of the protein, the amino acid residues which are present in the protein. Okay, so this is how a COSY spectra looks for a valine amino acid residue. So in this way, there are COSY peaks of all the different 20 amino acid residues. Okay, how the COSY spectra looks for each of the different amino acid residues. So now, what happens in case of COSY is that you can only this if this so only one bond J coupling can be observed, right? So your proton, if your amide proton is coupled to alpha carbon proton, only that information is provided by COSY. But as the bonds increases, like as the coupling or as the distance increases, that information is lost. So the J coupling or the J value decreases for longer distances. So it will not give you the, so your in a way your alpha or your amide proton is also coupled to the gamma proton, right? But that information is not cannot be provided by COSY experiment. It is provided by a TOCSY experiment. Okay, so because with the distance, the coupling, as the coupling constant value decreases, that particular peak will not be observed. Okay. So you will have a peak here, amide proton coupled to alpha proton, but you will not have a peak over here because of this. So so this is an important point that in COSY, cross peaks will not be observed for small J coupling. Okay, for small J coupling. So for example, this are your amide proton and your gamma proton. They are having very small J values. Okay, J coupling values. So as a result, we will not observe cross peaks for small J coupling. Okay, so cross peaks will only be observed for H beta, H gamma. So one bond coupling like H beta, H gamma, or two bond coupling. This CH two or three bond coupling when it is present, only that those cross peaks will be observed. Cross peaks will be observed for H beta, H gamma, but not for H gamma, or amide proton, gamma proton. These will not be observed. Okay. So another important point is that your information, the coupling information will only be present in a single amino acid residue. COSY will not provide you the information about the next amino acid residue, that whether this one, this proton, amide proton is connected to the I+1 amide proton. That information will not be provided by a COSY experiment. So COSY detects the J coupling networks between protons of amide proton, H alpha proton, H alpha proton, H beta proton, H beta, and H gamma proton. This creates a characteristic pattern of cross peaks. So for each amino acid, we will have a characteristic pattern of cross peaks. So in case of valine, the proton system can be defined by this amide coupled to alpha proton coupled to beta proton, and beta proton coupled to gamma 1 proton and gamma 2 proton. So COSY can therefore be used to identify individual spin system of amino acid residues. Okay. So COSY primarily identifies J. This is a very important point. So your COSY will only identify J coupling connections within a residue. Amide proton of one amino acid residue, that is, it is coupled to the alpha proton of that same amino acid residue. Okay. It will not give you information about the H alpha I plus 1 information. That will not, or your the proton which is present in your alpha carbon, whether it is coupled to the I plus 1 amide proton, that information will not, that cross peak will not be present in your COSY spectrum. Does not normally give a J coupling correlation as H alpha I to NH I plus 1 because the required coupling pathway is not strong enough. So as the distance increases, your coupling, J coupling value will decrease. So you will not have cross peaks. Okay. So now, if you, so what you were seeing is that in a COSY spectra, you were having the diagonal peaks and you are having the cross peaks. Now, if you zoom into the cross peaks and the diagonal peaks which are observed in your 2D COSY experiment, then you can further observe the what are the fine spectra which is present in the, what is the fine spectra present in your, present in this COSY experiment, right? So so this is actually a zoomed view, or if the resolution is good, then you can observe these fine structures which is present in your spectra. So basically your 1D spectrum will contain, if you have to a spin system, AX spin system. So AX spin system means what? That A is coupled to X, X is coupled to A. So for A, you will have a doublet, and for X also you will have a doublet, right? Similarly, in the F2 dimension, you have doublet of A, doublet of X. In the F1 dimension also you have doublet of A and doublet of X. Okay. So now, so your 1D spectrum contains two doublets. Okay. So the corresponding COSY spectrum will contain two diagonal peaks and two cross peaks. Okay, so these are your two diagonal peaks, this one and this one. These are your diagonal peaks. And these are your cross peaks, this one and this one. Okay, this and this are your cross peaks. So the diagonal, what is, so to do all those quantum mechanical calculations, quantum chemistry calculations, then you can like derive the cosine terms, sine terms, and why these are absorptive, dispersive. But what is, when you zoom into this fine structure, what is observed is that these diagonal peaks show dispersive line shape. So basically in this figure, what has been done, so this is your COSY. So this is a contour map, the contour plot. Okay. And if you look at these signals, at each of the signals, so along this line, along this line, along this line, and along this line, what is the type of frequencies present in each of these four lines? Those has been those frequencies have been shown in this right hand side figure. Okay. So your diagonal peaks show dispersive line shapes. Okay. So these ones, these diagonal peaks, if you see, these diagonal peaks have dispersive line shape, while the cross peaks will show you absorptive line shapes and characteristic positive and negative peaks. So these are your, so your cross peaks. What is seen is that these are your cross peaks. So your cross peaks will have absorptive line shape. Here also you can see that this one. This one, these are absorptive peaks with characteristic positive and negative component, minus, plus, plus, minus. While your diagonal peaks, these are having dispersive line shape. Okay. So these are your actually this and this, this and this. The difference is important because the The difference is important because the cross peaks are much easier to interpret quantitatively. Now, what happens, because so you have to understand that these cross peaks that we have, they are all absorptive. They are absorptive, and because of this absorptive nature, these cross peaks can be easily now, if there are absorptive signals, they can be easily resolved. Absorptive signals are easy to resolve, while this dispersive signals, they cannot be resolved. So that is why the cross peaks are easier to interpret quantitatively. So this is minus plus, minus plus, and for this again you have, here it is again reverse. It is so the fine structure of the cross peaks contain information about the J coupling. So depending on, like you were seeing the cross peaks, so these cross peaks are, so what is the separation between them, that provides information about the J coupling. Okay. So that provides information about J coupling. So the separation between the components can be used to determine the coupling constant. The diagonal peaks also contain coupling information, but their dispersive or complex line shapes make accurate extraction difficult. The COSY cross peaks can contain both positive and negative components. So one thing you have to understand over here is that the cross peaks are absorptive. They are absorptive with both, with positive and negative components. Okay, absorptive why? Because they are having cosine terms, and your diagonal peaks are dispersive. They are dispersive. Okay, and that is why, because of this diagonal peaks being dispersive, the J coupling information cannot be extracted from the diagonal peaks, while the cross peaks are absorptive, and therefore the separation between these fine structures can provide information about the J coupling between the two spins. Okay, if the coupling is very small, so what happens? One of the disadvantages that if the resolution is not very good, then what can happen? This plus minus minus plus, these can be if they are not very well resolved, then this plus minus can come together and they can lead to cancellation of the peaks. Okay, and as a result, the information will be lost. So if the coupling is very small, uh, so if the coupling is very small, so as was discussed also, like you will not observe cross peaks between H gamma and H alpha, right? Because that was resulting in four bond coupling, and this four bond coupling is so weak, this plus minus and minus plus gets cancelled. So so because these cross peaks contain both positive and negative components, so they can cancel each other. So these signals that you see, these are actually signals of the cross peaks. Okay, minus plus, minus plus, then plus minus, plus minus. Okay, antiphasor. So actually, this has been taken along this line. Okay, so this one, that is why you are getting antiphase absorptive, and this is in phase dispersive. So your diagonals will always give you dispersive signal. Okay, and again, when you are taking along this one, you will have this sign will be interchange, and you will get opposite antiphase absorptive and in phase dispersive. Now, another concept that was discussed was the double quantum filtered COSY experiment. So the COSY sequence is modified to select a particular coherence pathway. So during the initial part of the sequence, several coherence pathways can be generated. The DQF COSY sequence introduces a filtering step that selectively retains the double quantum coherence pathway. So this step that you see here, what is done here, a filtering step has been introduced so that only the double quantum coherence pathway is retained, and Then another pulse converts this DQC into single quantum. So then this T2 period, this DQC is converted into a single quantum coherence. And also another important point is that this DQF COSY experiment do not give signals of singlets. Okay, so like if you have water signal, that will give a very strong peak. So those signals are also signals. So say this signal, say a singlet. So a singlet has no J coupled partner, right? So therefore the singlet will only be present in the diagonal peak. It will not produce a cross peak. So in this DQF COSY, what is happening is basically this filtering process that is introduced, this 90 here in this one. After this 90 degree pulse, you are applying a filtering step. And due to this filtering step, what is happening? Only this double quantum, it will selectively retain the double quantum coherence pathway. And as a result, you will have both absorptive signals in both the cross peak and in the diagonal peak. Okay. And as a result, the resolution of the peaks that you get is they are quite well resolved compared to that of a COSY. Okay. And also the singlets are also eliminated because they are not J coupled partner. They do not have J coupled partner. So the singlet peak, if you have these signals, you can see here that they are even in the diagonal peaks, you can see they are very they are not well resolved. They are clumped together. Okay. So now answer this question. In a COSY spectrum, the fine structure in the diagonal peak have. What will be the answer for this question? So we saw that the diagonal peaks will have dispersive components, right? So it will be what? So they will have in-phase dispersive components. Okay. We saw this right now. You can see here plus, 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 plus. So that means that all the diagonal peaks are having in-phase dispersive components. In-phase dispersive components. And your cross peaks will have, it will have both they are all absorptive components, and it will have both antiphase and in-phase, right? Antiphase absorptive components and in-phase absorptive components. They are all absorptive. Your cross peaks are all absorptive with both antiphase and in-phase signal, while your diagonal peaks, they will always be in-phase dispersive components. So that is why answer is option C. Now in a 2D COSY spectrum of a proton, cross peaks are seen. So just now we understood this concept that within a 2D COSY spectrum, within an amino of a proton, cross peaks will only be seen within an amino acid, right? Answer is option A. We just now saw that because as your bonds increases, the J coupling weakens, and so we will not observe cross peaks for smaller J coupled spins. Okay, so we will only see cross peaks within an amino acid and not between near neighbor amino acids. Further amino acids also we will not obtain. So these informations are actually given by TOCSY. Between protons separated by more than 4 bonds, this information also cannot be provided by 2D COSY experiment. Okay. So in a 2D COSY spectrum, cross peaks are seen between. So we just now saw that 2D COSY spectrum, cross peaks will be seen between J coupled spin, isn't it? J coupled spin. COSY experiment will provide information about the J coupling between two spins. Okay. NOE provides information about dipolar coupled spin, but COSY experiment provides information about the J coupled spins. That is why your cross peaks we discussed just now, because your cross peaks are having absorptive signal, and from those signals we can actually get the information what is the coupling between the two spins. So we will look into this concept once we discuss the AX and AMX spin system. So we can solve this, right? So cross peaks for a two spin system in a 2D COSY spectrum will have cross peaks. So just now we saw that, right? Your COSY, your 2D COSY spectrum will have what? It will have diagonal peaks and cross peaks. So your cross peaks are having, while your diagonal peaks are having this type of signal, right? Sorry. So these are your diagonal peaks. So your diagonal peaks will have in phase dispersive signal. It will have in phase dispersive signals, and your cross peaks will have, what will your cross peaks have? Your cross peaks will have in phase, both in phase absorptive and antiphase absorptive signals. In phase absorptive signals and antiphase absorptive signals. Okay, in phase means plus and antiphase means minus. Okay, but they will all be absorptive signals because they are all cosine terms. And here your diagonal peaks are all sine terms. That is why you get dispersive signals. Basically, these are actually how you get these plus, minus, minus, plus. So these terms contribute to the plus and minus in phase absorptive signal. This is anti-phase absorptive signal. This is again anti-phase absorptive signal, and this is in-phase absorptive signal. So these terms give you these in phase the frequency that we saw. These are dispersive signals, or there can be absorptive signals. Okay, these are absorptive signals. These are dispersive signals. Okay, so now answer this question. In a cross peak of a 2D COSY spectrum, the line shapes along the F1 and F2 dimension will be. So the cross peaks will have what? So just now we discussed, right? So they are all absorptive line shapes. Okay, cross peaks will always have absorptive line shape. Absorptive line shape. Okay, they will have absorptive line shape, cross peaks with positive and negative component, and your diagonal peaks will have what? Dispersive line shapes. Dispersive line shapes, and they will all be in phase. Okay, in phase dispersive line shape. In phase dispersive line shape. That is why answer will be what for this question? It will be absorptive, 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 absorptive. Option D. Okay. Cross peaks will always give you absorptive line shape. Okay, but it can be in phase or anti phase, but it will always be absorptive. Okay, so now another concept that was discussed is that how do you increase the resolution in a 2D spectra? So when we are talking about resolution in 2D spectra, then we have to consider both the F1 dimension and the F2 dimension. Okay. So in general, what is the general rule? So as you increase the acquisition time, your resolution will improve. So your resolution is inversely proportional to the acquisition time. So the higher the acquisition time, your this distance between the two points in the FID, that will decrease. That means if the distance between these two points in the FID, if they decrease, it means that the resolution is improving. Okay, so so in general, it is what is done is that you have to increase the acquisition time. So in if when we are talking about the F2 dimension, in the F2 dimension you are collecting the FID, right? In the F2 dimension you collect the FID. You are collecting the FID. So when you collect the FID, we had learned that this T acquisition, it is equal to the separation between these two points. That is called as the dwell time. The separation dw and the number of points that we have collected, that is indicated by F2 between each point. So if you then you will have so increase the number of acquired data points and DW is the dwell time. Okay, so if you collect more number of data points, your acquisition time will improve, and as a result, your resolution will improve. But in F1 dimension, what has to be your increments? Like in the F1 dimension, we were collecting each FID with increase in with each increments. So if we take each increments as delta T1, and if we make if we make N1 such increments, then your T1 will be what? Your T1 will be N1 into delta T1. So N1 is what? It is the number of T1 increments that we have made, and delta T1 is increment between successive T1 values. Okay. So but the problem with F1 dimension is that if you increase the number of T1 increments, then your experiment time increases significantly. If you, for example, here, if you want to increase the resolution in the F2 dimension, so for example, you are collecting 2048 points. Now you are collecting 4096 points. So from 50 millisecond, it can go to 100 millisecond. Okay. But your experimental time will, so that is not much of a difference, right? But when you are increasing the T1 increments in the F1 dimension, then it can result in the experiment time getting much longer because of increments because of this T1 increments. Therefore, the F1 resolution must be optimized rather than increased indefinitely. So we cannot indefinitely increase the F1 or increase the T1 number of T1 increments. Okay, because that can result in longer total experimental time. Okay. So that is why we have to optimize this. So why F1 is important for COSY? Because we saw that in the F1 resolution actually carries the information about the J coupling, right? And if your F1 resolution is not if it is not well resolved, then what can happen? Your positive and negative peaks that were there in your cross peaks, they can overlap and they can cancel each other. Okay, in phase absorptive signal and your anti phase absorptive signal, they can cancel each other. So that is why it can result in poor resolution, resulting in signal cancellation. So the experimental time along T1 dimension has to be optimally adjusted. Why? Because we cannot indefinitely increase the number of increments, because if we indefinitely increase the number of increments, then it will result in much longer experimental time. So that is why we have to adjust that. So we can see only and another is that this F1 resolution is very important. So it has to be optimized. If you do not optimize it sufficiently, then your positive and negative peaks can cancel each other. So now it was mentioned in the lecture that around 512, this T1 is selected in a way that it is around 512. Now there were two cases that were discussed. So for a three-spin system, what will happen? So we looked into a two-spin system, AX. So your AX spin system will give you what? So if you look at the COSY spectrum of a AX spin system, so if this is your A, your F2 dimension will have frequency of A and frequency of X. Similarly, your F1 dimension will also have the frequency of spin A and frequency of spin X. So you will get a diagonal peak, right? This is the self that each spin is correlated to itself, autocorrelation. And A, if it is coupled to X, you will get a cross peak here. You will get a cross peak here. And A is also coupled to X, so you will get a cross peak over here. So these are your cross peak. Now, if you have a three-spin system, AMX spin system, now there can be two cases. Either your these three spins are linearly coupled, or they are coupled to each other, or they are in a way that they are triangularly coupled to each other. So this is a triangular coupling between the AMX spin system. So what will happen if they are linearly coupled? So if they are linearly coupled, then what will happen? So if you draw the 2D COSY spectra of a linearly coupled AMX spin system. So this is your A, this is M, this is X. So basically, here also you will have A, M, and X. Okay. So first of all, you will get the diagonal peak in the sense that A is coupled to A, M is coupled to M, X is coupled to X. Now A is also coupled to M, right? A is also coupled to M. So you will have a cross peak over here, right? Similarly, if you look at this axis, this M is coupled to A. So you will also have a cross peak here, right? Now A is not coupled to X, isn't it? A is not coupled to X. M is coupled to X. So you will not have a cross peak here. But now if you move further, this M is coupled to X, right? M is coupled to X. So you will have a cross peak here. And this M is coupled to X, so you will have a cross peak here. Okay. So this is how your 2D COSY of a linearly coupled AMX 3-spin system will look like. Okay, and also one more, how your 1D NMR spectra of this linearly coupled AMX spin system will look like. So your A is coupled to M. So M will result in splitting of the A spin into doublet, right? So you are getting a doublet of A. Now M is coupled to X and it is also coupled to A. So you will get doublet of a doublet for M, and for X you will get only a doublet. Okay. But if it is related, if each of the spins are coupled to each other, so A spin is coupled to M, and X is also coupled to A, M is also coupled to X, then what we will have? So when you draw the 2D COSY spectra of this AMX spin system, so first of all, you will have the diagonal peak, A, M, and X. Now A is coupled to M is coupled to A, so you have a cross peak coming over here. A is also coupled to X. You can see A is coupled to X as well, so you will have a cross peak over here. Now similarly, if we see this axis now, M is coupled to A, so you will get a cross peak over here, and X is coupled to M, so you have a X is coupled to A, so you have a cross peak here as well. X coupled to A. This is A coupled to M. Then X is coupled to M as well, so you get a cross peak here. Similarly, A is coupled to X, so you have a cross peak here, and M is coupled to X, so you have a cross peak here. So you will have all these different cross peaks. Okay. And the 1D spectra will give you doublets, doublet of doublet. X will also have doublet of doublet, M will also have doublet of doublet, A will also have doublet of doublet. So this was explained, right? Now, one other thing that was discussed is the active coupling and passive coupling. So now if we look at one of the cross peak, okay, suppose we are considering this cross peak. So what is this cross peak? This is the AX cross peak, AX cross peak. Or if we consider this AMX system, this is what? This is your MX. This is your MX cross peak. No, this is your AX cross peak, right? Because you have A over here. So this is your AM cross peak, right? Because your A is coupled to M, so this is your AM cross peak. Okay. So this is your MX cross peak. This is your AM cross peak. So if we look at the fine structure of the AM cross peak, then what will happen? So what is basically what you have to understand over here is that if we consider the AM cross peak, then the cross peak that is observed because of the coupling, that will be called as the active coupling, and the other peaks that we will get in the fine structure, they will be called as the passive coupling. Okay, so if we hear what is shown in the lecture was that we are considering only the AM cross peak. Okay, if we consider the AM cross peak, then JAM is the active coupling, while your JMX is the passive coupling. Okay, JMX is the passive coupling. So your AM, this is your M. Okay, so yeah, so how this peak splitting will occur? So your A is coupled to M, right? This system. So A is coupled to M, so M will result in splitting of this frequency of A into. So active coupling will always give you plus minus sign, plus and minus, right? And then if we consider this M, so M is coupled to A and it is also coupled to X. Now if we consider that JAM coupling is stronger than MX coupling. AM coupling is stronger than JMX coupling. Then what will happen? First of all, this frequency of M will split into, will split into two. That will, so this is active coupling. A is actively coupled to M, so it will give you a plus minus side, and then this X is also coupled to M, so this will result in passive coupling. So passive, this plus will be split into plus plus, and minus will be split into minus minus. Okay, so that is why you will have this type of your M. For your M, you will have this type of signal, plus plus minus minus, and for your A you will have plus minus signal. Now if you multiply these two, then what you will get? Plus plus will give you plus, plus plus again will give you plus, minus minus will give you plus, minus this minus plus will give you minus, and minus plus will give you minus, so you will get this type of signal. So basically you are having two components in the in the F1 dimension and four components in the F2 dimension. So 4 into 8, you will get 8 components. Okay. Now, if you consider that your AM coupling, JAM coupling is less than MX coupling, then what will happen? First of all, your M, your frequency M, it will be split into two. So it is actively coupled. So it will be split into a positive and negative signal. And then, so this is your JAM. And then this will again be split by the X pin into plus plus and minus minus. Okay, now because it is weakly coupled, this minus signal will come over here. So what you will be having here, if you try to plot the, so here if you try to plot the 2D spectra, then what you will get for A you have, so if you plot it, this A over here and M over here, for A you have what? So A is actively coupled to M, so you have this plus minus signal. Okay, and for M now you will have plus minus, plus minus, plus minus, plus minus, and when you multiply this, you will get plus minus, plus minus, plus plus plus, minus plus minus, and again you will have plus minus minus, minus minus plus, minus minus. So you will have this type of signal, plus minus, plus minus, minus plus, minus plus. Okay, so these are the two cases that were discussed actually. So for this AMX pin system, you can have two cases. Either your JM coupling is stronger than your MX coupling, or your JAM coupling is weaker than your MX coupling. Okay, so first of all, if we look at the splitting of spin A, then and if we consider the fine spectra, so here what we are doing, we are considering the fine peak of the AM cross peak, fine peak of the AM cross peak. So if we consider the fine peak of the AM cross peak, so if you are considering the fine peak of AM cross peak, then your JAM is the active coupling. Okay, so that is why your A frequency will be split into plus and minus. So this is your JAM, right? And then this will again be split into. So now AX will be passive coupling, right? So this will be JAX, right? This will be JAX. Now they are passive coupling, so this plus will be split into plus plus and minus will be split into minus minus. So now when you plot this, so basically your A will only give you plus plus, plus minus, right? A is only coupled to M, right? A is only coupled to M, so your A will only give you plus minus. So this will only give you plus minus, right? But for the M, when you consider M, so M is actively coupled to A, so that is why you will get plus minus, and then it is passively coupled to X. So this is your JAM and plus plus minus minus, minus minus. So you will get the plus plus minus minus. So this will give you plus plus, this type of signal. So these are the two cases that you will obtain. So this you will obtain when your JAM coupling is greater than JMX coupling, and this you will obtain when your JAM coupling is less than JMX coupling. Okay. So the fine structure in the AM crosspeak for a 3-spin AMX spin system. So for a linear AMX spin system, your A will have A is only coupled only to M, so it will be a doublet. M is coupled to both A and X, so you will get doublet of doublet. X is coupled only to M, so you will get a doublet. Now, if we consider the AM crosspeak, so your active coupling will be what? Your active coupling will be JAM. Okay. Now, passive coupling will be what? Passive coupling will be JMX. Okay. So if you consider this AMX spin system, and if you consider the fine structure that is present in the AM crosspeak, for example, then your M will be, and if you are considering only the AM crosspeak, if you are considering the AM crosspeak, then your JAM will be active coupling. Your JAM will be active coupling, while your JMX will be passive coupling. The reverse will be true if you are considering the MX crosspeak. Okay, vice versa. So active coupling will result in peak splitting into plus minus, and passive coupling will result in peak splitting into plus plus or minus minus. Okay, so this is the important point. Passive coupling result in peak splitting into plus plus and minus minus. Active coupling will result in plus minus peak splitting. So if your A dimension will have two components because A is only coupled to M. So A will only be split into plus and minus by the M spin, and your M will be split into doublet of a doublet. So it will have plus plus minus minus, or it will have plus minus depending on whether the MX coupling is stronger than the AM coupling, or whether it is vice versa. So okay, so similarly for the MX crosspeak, so if you are considering the MX crosspeak, your MX crosspeak is which one? So this is your MX crosspeak, this one is your actually MX crosspeak. This is your AM crosspeak, this is your MX crosspeak. MX crosspeak, if you consider MX crosspeak, then it will be active coupling. MX is the active coupling, while AM is the passive coupling. Okay, so active coupling will result in plus minus splitting, and passive coupling will result in plus plus and minus minus splitting. Okay. So now if we consider the three, if it is triangularly related, right? If it is triangularly related AMX spin system, then what will have? A will have doublet of doublet, M will have doublet of doublet, X will have doublet of doublet, right? So now there can be many cases. So if we are considering, for example, here the MX crosspeak has been considered. So if you consider the spin peak splitting of this spin X, then X will be split into. So if you are considering the MX spin MX crosspeak, then MX is actively coupled. So this new X will be split by this MX coupling into plus and minus. Then there will be passive coupling of the AX system. So it will be plus plus minus minus. So you are getting plus plus minus minus. For M, what will be then? So for if we consider the frequency of this M, peak splitting of this M spin, then again for MX it will be plus minus, and it will be plus plus minus minus. So as a result, when you multiply this plus plus minus minus and plus plus minus minus, you will get how many components? So you have 4 components here, 4 components here. So total you will have 16 components. Okay, so when you are considering the MX crosspeak, your MX is active coupling, AX is the passive coupling, and JAM is also the passive coupling. So for MX, it will be, so for this 3-spin system, now A is coupled to M, A is also coupled to X. So that is why AX will be, if you are considering MX crosspeak, then your MX is the active coupling, while other two are the passive coupling. If you are considering the AX crosspeak, then your AX is the active coupling, other two are the passive coupling. Now, another important point is that fine structure will depend on the active and passive coupling, right? Plus, plus you are seeing here, right? The fine structure, what is the, it is actually depending on which one is active coupling and which one is passive coupling. And the relative magnitude of the active and passive coupling. It will also depend on the peak appearances on the two sides of the diagonals will be if the peak appear. So if the two sides are different, so this was the case that was shown over here. So if the two sides are different, then again it will be, then it will again give you different types of fine peak. Okay, the peak appearance on the two sides of the diagonals will be different. So in a triangular AMX spin system, every cross peak will have what? So if it is a triangular spin system, so each one is coupled to the, each one is coupled to each other. So A is coupled to M, A is also coupled to X. Similarly, M is coupled to A, and each is coupled to the other two, right? So A will have a doublet of doublet, and M will have doublet of doublet, and X will also have doublet of doublet. So we were getting like for A, new A will be split into two, plus minus, and this will again be split into plus plus minus minus. Similarly, for if we consider X, then X will be split into plus minus, and this will again be split into plus plus minus minus. So when you multiply them 4 into 4, you will get how many components? 16 components. So answer is 16. If it is a triangular system, you will get how many components? 4 into 2, 8 components. Okay. So you can go through the explanation. It's the same thing. Okay. So now in a linear 3-spin AMX, in the cross peak between A and M spin, so the spin which were the cross peak between the two spins that we are considering, that will be the active coupling. So that is the main important thing. So if you are considering cross peak between A and M, then AM will be the active coupling basically. If you are considering the cross peak between the M and X, then JMX will be the active coupling. Okay. So linear 3-spin system. So if you are considering the AM cross peak, if you are considering the AM cross peak, then your JAM is the active coupling. If you are considering the MX cross peak, then you will not have AX cross peak, right? Because A and X are not coupled. MX cross peak, then your JMX will be the active coupling. Okay. Other will be passive coupling. Now in a 2D COSY spectrum for a 3-spin AMX system, the fine structure in cross peak, 3-spin AMX system. 3-spin AMX system, the fine structure. So that's now we learned this. So if it is a 3-spin AMX, it is determine the relative magnitude of the chemical shift, determine the relative magnitude of the active and passive coupling constant. So answer is option B. Just now we saw that it is determine the magnitude of the active and passive coupling constant. And it will also depend on the signs, right? We saw active coupling, and if it is active coupling, it will result in plus minus splitting. If it is passive coupling, it will result in plus plus or minus minus splitting. Answer is option B. In a 2D COSY spectrum for a linear AMX spin system with JAM greater than JMX, the M spin multiplicity will have the structure. So you have a AMX spin system, right? The M spin multiplicity will have structure. So if we consider M, okay, so M will be, and JAM is greater than, it has been said that JAM is greater than JMX. Okay, so first of all, this AM will result in splitting of this M spin into plus and minus. So this is JAM. And then again, there will be splitting of this passive splitting by this. So if we are considering A, for example, we are considering the AM cross peak, then this MX, JMX will be the passive coupling. Okay. So it will be plus plus and minus minus. So it will have a pattern like plus plus minus minus. If JAM greater than JMX, it will have plus plus minus minus. If it is the reverse, JAM less than JMX, then it will be plus minus plus minus. Okay, because this one will overlap with this one, and it will now have the minus over here. So you will get plus minus plus minus. Okay. So answer is basically option B. Now, the last two concepts that were discussed was the J-scaled COSY. So in J-scaled COSY, what we are doing is we are basically artificially increasing the apparent J coupling along the F1 dimension. So along this F1 dimension, so this example was given uracil of that of a uracil. So along the F1 dimension, you can see that the J that this J coupling in the F1 dimension is 4 times that of the J coupling in the F2 dimension. So that is what has been written. So as a result, the benefit that we are getting is that the minus and the separation between the negative component and the positive components are increasing. Okay. So we will get the disadvantage of the peaks cancelling each other, that is nullified by this type of pulse sequence, by this J-scaled COSY pulse sequence. So the actual molecular J coupling does not change the pulse sequence. So here what we have done is that we have the time that we have provided, that JF1 will be equal to alpha times J. And then there is again a spin echo sequence. Okay. So the important point is that the components will become farther apart, and the fine structures will become easier to resolve. So the cancellation is reduced. The actual molecular J coupling will not change. The pulse sequence makes the coupling appear larger in the F1 dimension. So you can see here that in the F1 dimension, the coupling appears larger. Now, the another concept that was discussed is that the CT COSY or the constant time COSY. So, in constant time COSY, what you are doing is that the time you are providing in the pulse sequence, the time that you are providing, that is constant, delta. Here, you are increasing that with increase in T1, your this T1 time is equal to alpha T1. So, it is the alpha can be anything, can be any integer 1, 2, 3, 4, 5, 30, 32. So, as you increase this alpha term, your J coupling J1 J will become enlarged. Okay. But here the total evolution time, your evolution time is constant. Yeah, it is so in J scale COSY, your evolution time was proportional to T1. Okay, it is alpha times T1. But in your constant time COSY, your total evolution period is constant. It is delta. It is delta. So total evolution period is equal to delta. So as a result, what is happening is that as T1 increases, the tau decreases. So this is constant. So because this delta is constant, if the T1 increases, your tau has to decrease, right? If you have to keep this delta constant. Okay. So therefore, J evolution is independent of T1. Only evolution that depends on T1 is encoded into the F1 dimension. So there's no J coupling information appears along the F1. So in the F1 dimension, you do not get any J coupling information, and the J splitting is removed along F1. Coupling information remains along F2. Along the F2, your coupling information will. So main advantage is that your overlapping cross peaks can become separated and easier to identify. So here you can see that the peaks were overlapping with each other, while in the constant time COSY, the peaks are now better resolved and they are separated. Okay, so they become easier to. So here the main point is that the J evolution is not dependent on T1, unlike the J scale COSY experiment. Okay. Now, last thing that we discussed that we have to that is left is the total correlation spectroscopy. In the total correlation spectroscopy, the important point is that you will not only obtain information within an amino acid, it will also obtain information between neighboring amino acid. Okay, so if we consider a linear spin system like A, M, X, Y, the COSY will only give you information of whether A is coupled to M, or M is coupled to X, X is coupled to Y. But your TOCSY can give you information about all these, whether A is coupled to M, it is also coupled to X, it is also coupled to Y. Okay, so the entire information can be obtained. So the TOCSY transfers magnetization through coupling network A to M to X to Y, and therefore the information is relayed through intermediate spins. Okay. So the entire J-couple spin information. So TOCSY show correlation across the entire J-couple spin system. So this is important. Now this was discovered. So you have the mixing time that you provide, that is having complex magnetization transfer sequences, and as a result you can get a relay of the entire J-couple spin system information can be obtained in case of a TOCSY when you do a total correlation spectroscopy experiment. Okay, and some of the important points that were discussed, that advantage of TOCSY is that unlike COSY, you have both plus and minus component, right? Your cross peaks were having both in phase absorptive components and anti-phase absorptive components, while TOCSY has what it has it will only have in phase cross peaks, it will only have in phase absorptive cross peaks, and as a result, the advantage is that there will be the problem of signal cancellation will be removed. We will get better peak intensity, we will get cleaner spectra and better apparent resolution. So another important point is that if there are two spins M and M dash, and if they are having the same chemical shift, so a COSY spectrum will only suggest whether AMX might suggest AMX, right? So if this is like M and M dash is having the same chemical shift, it will suggest that they are coupled to each other. But the actual situation can be that A is coupled to M and separately M dash is coupled to X. So if M and M dash overlap, the COSY can create an ambiguous connectivity interpretation. But if you perform a TOCSY now, so for example, you have AM separate and M dash is separately coupled to X, your COSY information will provide the information that A is coupled to M and M is coupled to X, but it will not be able to differentiate that whether this A is coupled to M, but M dash is not coupled to A. It is coupled to X. That information will be provided when you do a TOCSY, because since your A is not coupled to X, you will not get a cross peak for AX. Okay, so it will, if it is AMX only, then this AX cross peak will be obtained in TOCSY. If it is not present, then you will not get a cross peak. So you will understand this M and M dash are two different. They are two different spins and they are not correlated to each other. Okay. So that was all for. So this is one last question. Let's solve it. In a constant time COSY experiment, splitting is what? Splitting is removed along the F1 axis. Just now we saw that in the F1 axis, you have a constant time that is delta, right? So the splitting is removed along the F1 axis because your F1, this J coupling, that information was present in the F1 axis and that is removed. So answer is option A. So in a constant time COSY, the total evolution period is kept constant. As a result, the J coupling evolution becomes independent of T1 increment and therefore does not get encoded in the F1 dimension. So F1 axis J splitting is removed and F2 axis J splitting is retained. Omega 1 decoupled flows. The answer is option A. Okay. So that was all for today. So in this lecture, we discussed what all concepts we discussed for in this lecture. So we discussed the concept of 2D NMR. So starting from the four periods that are there in 2D NMR: preparation, evolution time, mixing time, and then the T2 period where the actual FID is collected. Then we talked about the different 2D experiments that are performed. So 2D J resolved experiment, and then we talked about the COSY experiment. So in COSY, we discussed about two spin system, AX spin system. We will get cross peaks, and your COSY gives you information about J coupling between the spin system. Then we also looked into the three spin system, AMX linear spin system and AMX triangular spin system. And also we discussed about TOCSY and the two other two methods of COSY, that is DQF COSY and constant time COSY. These two COSY experiments we quickly revise. So all these concepts were discussed in the lecture. So we tried to solve a few questions so that the concepts are even more clear. We'll stop for today and we can take any other questions or any doubts you have in the next session.